Thank you so much for that introduction, and thank you, Sarah and Virgil and everyone, for inviting me to speak with you this evening. I'm really excited to chat about some of the research that I've been working on in collaboration with all of you folks at Endangered Wolf Center on red wolves. So, as Sarah mentioned, I am coming to you from the Smithsonian National Zoo and Conservation Biology Institute. Mouthful. Okay, it's all downhill from here on the talk. So we are out in Front Royal, Virginia. This picture got a little bit chunky on the view screen here, but we are a research facility park focused on wildlife conservation, and very similar to the goals that everyone here at probably in this room have focused toward endangered species conservation. We take a lot of different avenues to reach our conservation goals. So some of the pillars of the research going on out at NZCBI include research in the field, satellite tracking of animals, their movement, the types of habitats that they occupy, educational outreach programs, zoo husbandry research, studying animal health, behavior, reproduction, and also participating in breeding population breeding efforts. And then the part that I do mostly is the laboratory work. So I'm out there underneath the microscopes, studying cells, understanding the reproduction of our species of interest, and developing assisted reproductive technologies that can support species conservation in these other two fields as well. So what am I talking about today? As Sarah mentioned, I'm mostly talking about sperm. So sperm can sum up this whole slide as per the picture. So I'm going to give a little history of red wolves for those of you who may not be familiar. Hopefully you are familiar since you are attending this talk and are interested in canid conservation. And then talk a little bit about some of the reproductive research, again, mostly focused on sperm. Karen will be very happy about that, that we've been doing with the species towards the future use of cryopreserved sperm artificial insemination, and then some of the work in ovarian and testicular tissue preservation and AI timing. So I hope this is a familiar story with some folks, but I'm going to do a really, really brief history of America's wolf, the red wolf. So this is a species of canid that is native to the United States. It used to exist all across the southeastern United States, anywhere these hashtaggy lines are on the map. Unfortunately, back in the early pre-1970s, there were some very aggressive predator control programs by US government and folks living in the areas where these wolves were, and also a secondary hit of coyote populations expanding across the United States from the West Coast to the East Coast, that when they hit those small populations of red wolves ended up interbreeding in some cases. And where we ended up having was a huge reduction in the number of wolves on the landscape. And ultimately, we ended up with only a couple hundred wolves or red wolf hybrids in the Texas, Louisiana area, where that red dot is. So in 1973, the Endangered Species Act was passed, and U.S. Fish and Wildlife Service decided that in order to save this species, we actually needed to bring it into captivity because it was not going to exist on the landscape in the current state. So all the wolves, remaining wolves, were captured and assessed to determine how much of red wolf they were, and brought 14 founder red wolves into a captive breeding population at primarily Point Defiance Zoo and Aquarium up in Tacoma, Washington. And then started this really expansive, that's lasted now for decades, captive breeding program. So we have the species survival plan that has managed the breeding efforts of this population in captivity since the 1980s. 1987 started the Red Wolf Recovery Program, where we actually started reintroducing some of these captive bred wolves back into the landscape in a location that's indicated in the blue on the map, which is the Alligator River National Wildlife Refuge. And at the time, there were no coyotes in that area. They hadn't reached the coast yet, so the chances of them not being able to find a mate and then deciding to pick a coyote instead because that's all they had were very slim since there were no coyotes there. And had a very aggressive reintroduction campaign that actually did quite well. So by 2011, there were over 100 wolves on the landscape, and they were actually mating, producing wild-born offspring, and for all intents and purposes thriving, and it was a successful recovery program. Unfortunately, the reintroductions kind of trickled down in the late, or the second part after the 2009 period. And we also started seeing a lot of the same problems that the red wolf populations had encountered back in the 1970s. That was the coyote populations had reached that population in North Carolina, and there's a bug. There was a lot of human conflict with the red wolves. So some of it was direct persecution again, but some of it was also road traffic accidents and stuff that were really affecting red wolf populations and are still a problem today. But in happier news, because I want to keep on the positive side of all these things, reintroductions of the red wolf have been reinitiated. That's a lot of re's, but reinitiated red wolf reintroductions as of 2021. And we are taking advantage of the fantastic community of institutions that are involved in this red wolf SSP and SAFE program, the Saving Animals from Extinction, and have been actively breeding and managing the genetics of this captive population for the past 30, 40 years. And so we actually do, although there are only 20 or so wolves right now in North Carolina, we have over 200 wolves in our SAFE institutions that are being actively bred and managed and are a great source for our future reintroductions. So if you're not familiar with the Association of Zoos and Aquaria's SAFE program, this is the goal for the red wolf SAFE, is to maintain that healthy and viable population of red wolves under human care for that later now reintroduction efforts, also to improve the education and awareness efforts, and to support research to aid in species recovery. So this little last bit, and also I guess that first bit, is mostly what I focus on, although tonight I'm doing the middle hat here of education and awareness. So yeah, I'm a SAFE ambassador here now. So my goal, and this is the goal of most of us in the field in general, is to have every individual's genetics represented in the next generation. When we have these really small populations of animals, if you end up losing one or two of them that they don't breed, and then their genetics aren't in the next generation, you've lost, with 14 founders, if you lose two, that's actually a huge percentage of the genetics of your population that you have. So when we start losing those genetics, we start losing the animal's ability potentially to respond to health threats. They may also be more susceptible to disease. And so we want to make sure that they are genetically healthy so that they can be physically healthy and thrive. So ideally with this, we have everyone just breed naturally. That would be awesome. Unfortunately, this has got a lot of challenges related to it. As I said, we have these 49 institutions across the United States. Sometimes when we're trying to match a pair of wolves that are the best genetic match, you'll have a female in California and a male in Florida. Someone's got to move for that romantic relationship to work. And so that can be quite challenging. And sometimes we make these pairs and they normally would not swipe right on each other if they met in person. There's behavioral incompatibilities, right? So we can't always predict that. We all, as the zoo community and a lot of the animal care keepers, do a fantastic job at bringing up the behavioral of the animals and when we try to do these matching things. But if we're going based on genetics, maybe those are your only options. So these geographical distances can be challenges, behavior can be challenges, and sometimes we unfortunately have animals that pass away before they're able to reproduce. If they don't get a chance to breed soon enough or something goes wrong, we don't currently have a lot of methods to rescue their genetics. And that's where some of these assisted reproductive technologies that I'm interested in that I'm working on are coming in. So one of the other challenges we also have is even when we do pair these wolves and they get along, based on our historic records of the red wolves from back in the 1980s, our pairs only produce offspring about 25% of the time. So 1 in 4 of these paired couples will actually produce pups. And so this is not sufficient for what we want to do, both in terms of the genetic viability of the captive population and also for our current release goals, because if only 1 in 4 are producing, we don't have a lot of pups that we can then send out into the wild for their reintroductions. So this is something we really need to be increasing our breeding efforts as much as we can. So some of the reproductive technologies to start addressing some of these challenges that we are having. Throwing up here, I'm not sure if folks are too familiar with them, but I think they're gaining a little more traction in the human fertility preservation field. So sperm banks, probably fairly common knowledge, the idea of preserving those genetics in liquid nitrogen and cryopreserving them. This would allow us to move the genes around rather than moving the animals, and also to have those in storage for if we need those genes later on. Similarly with egg banking, egg cryopreservation, artificial insemination, this is how we'd actually then use those banked sperm. I consider this the turkey baster method. So unlike in vitro fertilization where you're putting your egg and sperm in a petri dish together and producing an embryo, we're just would be using the sperm side. But that, again, an IVF or in vitro fertilization would be another technique that we might be interested in the future. And then some other things like pregnancy detection would help in the management of our species. We don't currently have a great pregnancy detection test for canids, and I'll go into this a little bit more in some of the future slides. And like I said, that issue of if we have an animal that dies before they're able to reproduce or dies outside of the breeding season where they don't have mature sperm and eggs that we can use for our banking, options to preserve the genetics through the freezing of ovarian or testicular tissue would be really useful. And this is actually something that's also increasing in human fertility clinics as well. Oftentimes you'll have women going in for cancer therapies, and the therapies themselves will destroy a lot of the eggs in the ovary. So they will take pieces of ovarian tissue and cryopreserve them and then either transplant them later or hopefully rescue those eggs and use them for in vitro fertilization. So some of these technologies are pretty well established in humans. We just don't have them worked out fully for canids, in part because we don't fully understand all the reproductive biology. We need to really understand the system before we're able to develop the techniques. I mentioned contraceptives here. I don't really work too much in this field, but it is one of the goals of the Red Wolf Reproduction Program is to optimize our contraceptives. Basically as another way to manage the population, because again, if you had 14 founders and one female or male pair produced 20 pups, you maybe don't want her to be or him to be producing anymore, and you want to give the others a chance to do it. And so this might be a way to manage the individuals in your institution without having to spay or neuter. So speaking mostly today about cryo-banking. I think this slide is kind of a little bit late now that I'm thinking about it in retrospect. But what I'm talking about here is the process of cooling and storing cells, tissues, organs at very, very low or freezing temperatures down to negative 196 degrees Celsius so that they are just basically in stasis. So they can hold there for years or decades until you take them out and warm them back up, and they should revive and for future use, also known as cryopreservation. And this has been very successful. Yes, I had to homage. Clearly been very successful and has a lot of potential for our endangered species. So the Mexican wolf pup-born here from cryopreserved sperm is a great success story from Endangered Wolf Center. So kudos, everyone. And then we've also had some stories where we have similarly 30-year-old cryopreserved sperm from the black-footed ferret that produce offspring, and more recently a black-footed ferret that was cloned from cryopreserved skin tissue from one of a potential founder line of black-footed ferret as well. So these are options to maybe reintroduce some of the genetics that were lost over the years of captive breeding, or at least bring it back up to the levels where it was before a lot of these generations have passed. All right, so the goal. I'm not going to repeat that, that's too much. So back onto red wolf sperm. So this is not what red wolf sperm look like if you just straight up look at them. This is the glamour shots of these guys. These are actually some sperm that we had cryopreserved and warmed and then fixed them to look at the sperm morphology and actually how some of their expression levels are with some specific proteins. But what I have here is the sperm head is in, do I have a mouse here? No, I do not. Sperm head is fluorescently dyed in blue, which is binding to the DNA in the sperm head. I have the sperm membranes, including this very important membrane that's on the head of the sperm that is needed for it to bind to the egg in green. And then in red, I actually have some proteins that are showing active proteins that are part of the process for sperm actually being able to gain the ability to fertilize an egg. Because they're not necessarily immediately able to fertilize right after ejaculation. They kind of have to go through a second puberty, as it were, as they're traveling through the female reproductive tract. So when I started working with red wolves back in 2017, not as long as some of the folks in this room, but I was really interested in, there's been a ton of amazing research over the last years in fine-tuning the red wolf sperm cryopreservation protocol. We've got bank samples in liquid nitrogen from 1984, I think. So there are some really, really old historic samples of red wolf sperm that are just waiting to be artificially inseminated into a female somewhere. So there's been a lot of optimization of this protocol, but what a lot of the studies have shown, and I know I've thrown a lot of graphs up there, but we'll focus on this one at the top left-hand corner for a moment. You'll have pretty good sperm motility. That's the y-axis here at the beginning. Most, like 80% of the cells are moving, alive, pretty happy. And then once we start cooling them down to cryopreserve, you end up losing motility over time. So they're maybe dying, or they're at least not motile, so they're not able to, if you use them in artificial insemination, travel up the female reproductive tract and reach the egg if they're not motile. And what we saw a lot of times is you'll have this drop from cryopreservation in any species, even the ones that have been really extensively studied, like humans and mice and cows, because cryopreservation, you're freezing that cell. It's going to negative 196 degrees Celsius. That's a lot of stress. I don't like the cold temperatures. I can't deal with 0, so I can't even imagine. So this drop in motility, maybe that's unavoidable. But what we would also see is that after thawing the cells, if you continue to incubate them in like an incubator for a couple hours, by about 4 hours, you've dropped actually down from your 50% motile to maybe 10 or 20% motile. And similarly with another study, they did some different stats on this one, but you saw that precipitous drop in motility over the first 4 hours of incubation. And so when I saw this, I was like, well, that's not great, because I came from a background of working with domestic dogs and working and developing in vitro fertilization in domestic dogs. And what we would do to put that dog egg and the dog sperm in together in a petri dish is we'd let them sit there for the sperm to fertilize for about 12 hours or 14 hours. So even if we were doing something like in vitro fertilization, where the sperm doesn't have to figure out where to go very far, it's in that much of a drop, 4 hours probably would not be enough. And so this drop in motility, I was like, we need to figure out what's going on here and try to mitigate this. Because if this is actually an indicator that the sperm was not cryopreserved, was crashing after cryopreservation, then it's probably not going to successfully be able to artificially inseminate. Probably not IVF either, but it could also be that we are not handling the sperm appropriately after thawing it. It could be that the culture media we stuck it in to incubate it for these 4 hours is just not good for the red wolf sperm. So what we did as the very first thing was checked how our red wolf sperm handled fresh conditions. So without freezing it at all, we took red wolf sperm, collected through ejaculation, just like we did here yesterday, washed it out, and then incubated it in a couple different solutions, incubation solutions that are used in a variety of different species. These are all the names. It doesn't matter what they are. It's just abbreviations, but they're all developed for either embryo culture or sperm handling or IVF in a bunch of different species. So we basically grabbed a bunch of the different culture medias and tested them in fresh red wolf sperm to see how well they survived. And what we found, again, time on the x-axis, percent motility on the y-axis, fresh sperm, was that there was a very significant difference based on the handling media we used. If we use the most basic one, which actually is the most similar to those previous studies, it's almost like a saline base, like a salt solution, but no other additives. Up here, a little bit of sugar. We actually saw that the sperm followed that exact same trend. So 50%, actually this was actually quite bad for the fresh sperm, it didn't like it at all, and then dropping in 4 hours to 10 or 20%. But when we maintained it in these two media, the purpley one and the blue, which are canine capacitation media, this was designed for dog sperm handling, actually. And this one's North Carolina State University, it's actually in CSU, was really, really great for the red wolf sperm. And actually, these are the two media that we used for domestic dog in vitro fertilization, and we're able to produce puppies for the first time. So these media that are great for the domestic dog also appear to be doing quite well for the red wolf sperm, which was very promising. So the next thing that we've done is then see, okay, these are allowing for the survival of fresh sperm. This appears to be a much better handling method. Now, what if we freeze sperm and then use this for the incubation post-thaw? Do we still see that drop in motility? We do see a little bit of a drop, but, well, is this now you're not gonna work? We had such a good run. Oh my gosh, you totally worked a minute ago. There, that one works. Aren't they cute? They're adorable. Oh my gosh. Oh, there we go. Okay. So these are both the same individual. We froze his sperm. We thawed it. We actually froze it in two different methods, but they're pretty much the same for this. And then 6 hours of incubation later, we checked the motility. And we actually still have a decent amount of motility. Karen, you agree? It's pretty solid, right? Not too bad. So definitely not a 10 to 20% motile sample. These guys are doing pretty well. So, and we actually had some samples that survived. We had 10 or 20% motility at the 18-hour mark. So in my mind, this means, okay, our cryopreservation protocol is solid. The sperm are surviving. They are actually showing signs of that. Remember I said that second puberty where they were getting ready to fertilize? They were showing signs that they were developing that in this incubation system. So these sperm, I feel like, are very, very well suited for in vitro fertilization or artificial insemination. So that's super exciting. Oops. Now they all videos go. It's not enough for me, though. So I want to see how we can further optimize the sperm. And the reason for that is this sperm that I just froze and thawed and showed you the day before are using the protocol that folks have been optimizing for 30 years, doing these piecewise, stepwise meals. So, standing on the shoulders of giants, this protocol is great. I really, really would like to use for artificial insemination the sperm that were frozen back in 1980, or back in 1990 before this happened. And those were not in those fully optimized conditions, so I'm not expecting them to be super great. Now we have some data from the post-thaw on those, but it's not something that we've checked up on over the years because it's a very valuable resource. So we basically need to have all of our ducks in a row before we even start thinking about going into those samples. So how can we make sure that our post-thaw sperm are just really 100% there? So what I've been interested in the last couple of years has been trying to make our post-thaw culture conditions as supportive of cryopreservation and maybe as naturalistic as possible so that they are in the happy environment that they would be comfortable in to get ready to fertilize. And one of the things that we've been looking at are these things called extracellular vesicles. And this is where I start throwing a bunch of large words at you guys, so just blink furiously if you're not following and I'll try to get the message. So these are, I actually think of them as cellular text messages, but I mean cell, not cell. So these are little membrane enclosed nanoparticles that are shed from all cells. They're little, I think, blebs of membrane, but inside these sphere membrane shells are proteins, RNAs, DNAs that are packaged and emitted from the cells that then go into the space between cells, they get into circulation, and they end up binding to and being internalized by the recipient cells, which then read that message of protein, RNA, DNA, et cetera, and will actually change their functions based on that. So this has actually been some really crazy, interesting work in the cancer fields done on extracellular vesicles because they found tumors will shed these and actually will get into circulation, and sometimes they will go to different parts of the body and prime those cells in different parts of the body for metastasis. So it'll actually make them more susceptible for the cancer spreading. It's sending a forward notice party through these extracellular vesicles. That part is a super scary thing, but this is something that's happening across all cells. This is part of how they're communicating with each other. And so this is really interesting in terms of the reproduction side, because there have been some great studies in the recent years showing that the female reproductive tract, or even the male reproductive tract, will secrete these little extracellular vesicles and communicate with the sperm and the eggs. And a lot of times they're communicating to support sperm or egg function. So you'll have improved sperm motility or improved embryo production in the presence of these extracellular vesicles. So this is a study actually published at SCBI by another postdoc in our lab who took extracellular vesicles from the fluids of cat oviducts. For some reason in animals we call the fallopian tube an oviduct, and in humans it's a fallopian tube, so it's the same thing. Oviduct makes more sense to me because it's where the ovary releases the egg into, and then the sperm come up and meet it, and that's where fertilization happens. So ova is an egg duct, oviduct. So I will use that probably for the rest of the speech. But she took the fluids from this and isolated all these little extracellular vesicles and then co-incubated them with domestic cat sperm. And what she found was that when she dyed these little vesicles red and then co-incubated them, they ended up binding to the sperm head. You can see where the yellow arrow is, and part of the tail. And when she looked at it with electron micrographs, you actually saw that some of these vesicles, this is really, really tiny. This is the sperm head here, zoomed in like crazy. We're actually binding to the sperm head, binding to the sperm tail, and fusing and releasing those cargo, those proteins and RNAs, into the sperm cell. And when she then used these sperm for in vitro fertilization, she had much higher rates. So the green here is the ones that were co-incubated with the extracellular vesicles, and the blue is a control. Much higher rates of embryos being produced, much higher rates of 8-cell, so more advanced stages of embryos being produced. And then the blastocyst stage is right before implantation happens, and she saw much higher rates of that as well. So something in these extracellular vesicles shed by the cat oviduct was actually changing sperm function and improving sperm function and fertilizing capacity. So this is kind of how we were seeing it, is that these little vesicles are being shed by the oviduct, and normally the sperm would actually be coming up from the uterus into the oviduct, meeting the egg, and these are actually what these vesicles look like in real life, zoomed in like crazy. They're these little beads, and they kind of look donut-shaped because in the micrograph they have to sort of dehydrate. But center filled with gooey proteins that are then being passed along to our sperm and eggs. So what has been found out in some other species, a lot of the work in the cows actually in this field, have actually shown that, this is not just in cats. Like, this is pretty broad effect where these extracellular vesicles are improving sperm egg function, right? Work in the cow has actually shown that depending on the stage of the reproductive cycle, the molecules that are packaged in these extracellular vesicles is dramatically different. So if the cow is in its breeding season, about to ovulate, it might have a lot more messages in these extracellular vesicles that are related to the cell cycle, like promoting cell divisions in an embryo. But if it's sometime earlier in the cycle where the animal is not actually about to ovulate, there's no egg around, the messaging will be completely different, which would make sense, but it does appear that it's hormone-based as to what the oviduct is actually secreting. So what we did with the red wolf sperm, going back to the red wolf sperm, what the application of this is we wanted to see if we can see the same effect in terms of promoting the sperm function using oviductal extracellular vesicles. Now, we were not collecting these from red wolves because none of the red wolves wanted to donate their oviducts for some reason. I don't know. But what we did do is get some dog ovaries and uteri from local spay and neuter clinics. And we're able to identify animals that were right around this stage of the reproductive cycle. Now, I know this looks like a crazy graph. This is the hormone reproductive cycle of a domestic dog. So over on the x-axis, we have days. So for like months worth of time. And then we have two hormones that you're maybe familiar with for human reproduction, estrogen and progesterone. So basically what's happening is that as an egg is growing and about to be ovulated, that's called the follicular phase here, and eventually they'll start producing estrogen, and then ovulation will happen in here, and then the ovary will actually heal back over and start producing progesterone that will maintain the pregnancy. So these are our two reproductive hormones for the female that we're focused on. And this part where they're producing progesterone is called the luteal phase. So what we did was we focused in on right around the time of ovulation and broke this down into like the egg is almost ovulated, but not quite. The egg is just about ovulated. Ovulation just happened, and then a few days later, and focused in on getting dog reproductive tracts that were in these discrete stages and looked at how they affected post-thaw red wolf sperm function. So these are the data from that study. We have the period of time on the x-axis, and then we have the percent motility on the y-axis. And the purple line is our fresh control from these animals. So we saw that nice maintenance of them for at least 4 hours, and then by the 18 hours, they had kind of crashed. But what we found was that when we supplemented the EVs, so hopefully these colors aren't too bad, but the orange, pink, blue, and green are the different stages of the extracellular vesicles, we actually were able to improve post-thaw motility, shift it up about 20% compared to the sperm that were not supplemented with extracellular vesicles. So it's not a golden pill. It didn't bring them back up to fresh values, but it was a good, we got a bunch more motility out of it, particularly in this 6 to 10 hour range, which could be really important for actually getting sperm that are able to do something like in vitro fertilization, or maybe even just prime them so that when you then use them for AI, they've already been exposed to the best conditions possible. So exciting about this, but we still then don't know exactly what's happening, right? So we've then supplemented these vesicles. We know that they have all these molecules and these proteins and RNAs that are doing something, but we don't actually know what those proteins are. So we actually sent out these vesicles for protein analysis and looked at we got a list of 1,000 proteins that were present in these vesicles. So that's just the one compartment. That's a lot of things to do. So this looks messy, but it could have been worse. I could have shown you something way worse. So basically what we did is then took that list and figured out, okay, are there like groups of them that are associated with the same functions? Are all these proteins involved in metabolism, or is there a large chunk of them involved in something like fertilization? And we did actually have a large cohort of proteins in these extracellular vesicles that were identified as being associated with sperm and egg recognition. So that might very well explain why for that cat project we saw improved fertilization, because if they were delivering these pro-sperm egg recognition proteins, maybe that helped them meet each other, right? We also saw a large cohort of proteins that were involved in metabolism. So glycolysis was a huge group of proteins in that, and that may explain why we saw improved motility in the sperm over this incubation period, if they were basically, these vesicles were delivering some of the machinery that the sperm might need to be maintained a little bit longer. So lots of different things to go through. The one that I will point out, just because I'm really interested in it for application to other assisted reproductive technologies, is heat shock or chaperone proteins. And this is something that has come up on the list of a lot of other studies where I've been working in like cows and horses and stuff, looking at extracellular vesicles. Chaperone proteins are these proteins that are like helping other proteins in the cell. So I think of it as like they're the assistants when things start going awry, and particularly in situations where the cell is stressed. And this is why they're called heat shock proteins. So if the cell is exposed to really warm temperatures, these chaperones will come into play because you actually have a situation where, I don't know if you guys are familiar with this. This is what I always think about. So your cell is doing normal functions. It's producing proteins that are doing their jobs. So these are the proteins on your conveyor belt that are being produced by the cell. Something stressful happens and there's a backup in your assembly line for making proteins and sending them out to do their jobs. So then you end up having these weird shaped chocolates everywhere. They're not functional and they're mucking up the system because they're clomming onto each other, and maybe you have someone here doing disaster things in their supporting role. And the chaperone proteins, what they're doing is identifying, or are supposed to be identifying, the faulty things that are being produced by the cell and either fixing them or targeting them for destruction or consumption in this particular one. So they're basically regulating this to make sure that you don't have a giant disaster train wreck thing at the end of the line, right? And they're really coming into play when there's a stressful event like heat shock or stress from cryopreservation. And so these proteins have been, these family of proteins and some specific proteins in this family have been identified in sperm that survive cryopreservation better, and have also been supplemented to sperm in some cases and improve sperm motility and ability to withstand cryopreservation. And so again, like I'm saying, relating it to the red wolf thing, this would be interesting, either whether it's coming from an extracellular vesicle or being supplemented to our sperm in the thawing process as a way to hopefully maximize the post-thaw sperm's recovery and ability to then fertilize. So I know we're getting really into the details, but hopefully I Love Lucy helps with that. So our next questions here are trying to figure out what we can take with this next step. So we do also see over the last couple of years that we have some individuals that their sperm freezes really, really well. Actually, that 6-hour one that I showed you was kind of one of the gold star wolves earlier, because his sperm lived for like 48 hours post-thaw. It was insane. So some sperm, great. From day to day, though, we actually will have some samples maybe that don't do as well. So you'll have some ejaculates that are just more sensitive to cryopreservation. They crash. And that's not ideal when we're trying to make sure that every individual's sample is banked, right? So we thought we'd take our background with these sperm proteins and what we've been doing with their incubation to better understand what's happening to the sperm during the cryopreservation process. Are they getting damaged? Are they losing proteins that might be valuable? Can we supplement them back through extracellular vesicles or through protein supplementation? And can we specifically focus on ones that are cryoprotective to support these post-thaw? Again, to try to take advantage of that bank that we have of samples in the future. So I threw these pictures in. You have no reference for what they are, but the sperm actually are loaded into, in this particular image on the top, I think this is some sort of ungulate species because this is a lot of sperm. But this is how we freeze sperm. So we either load it into these really, really thin straws and then freeze it over the vapor of liquid nitrogen, and this is what I was doing yesterday, was actually putting our sperm, and they're yellow not because they're yellow. I mess with your colors the whole time. They're actually in a solution that contains egg yolk, which helps to support the sperm's survival during freezing. So they're in little droplets here that we use to freeze those. So also cute. This is one of the things we're doing. We're actually looking at the protein composition of red wolf sperm before and after cryopreservation to see how the freezing process, the damage of the freezing process is damaging or not to the sperm, and identify hopefully candidates that we can use to mitigate those changes. And then what we're also doing is looking at the seminal plasma. So when we get an ejaculate of sperm, it's the semen has got the sperm and seminal plasma in it. We've been taking out the seminal plasma and looking at the proteins in that as well, because there has been some really compelling evidence from agricultural species, like sheep, that adding the seminal plasma from a male that's cryopreserved really well to a male that does not typically cryopreserve well will actually rescue the sperm post-thaw. So this is figure here, again, same time frame of post-thaw on the x-axis and the motility of the sperm. This line right here was a good sperm cryopreserved words. A ram whose sperm froze really well and survived post-thaw. And down at the bottom is one that was consistently a low performer, low cryopreserver. And the low cryopreserver, when they added the high cryopreserver seminal plasma, partially rescued the effect. So we don't know whether this is through the extracellular vesicles that we're talking about, although it could very well be. but there's certainly something in that seminal plasma, likely some sort of protein that is facilitating this cryosurvival, and that could be very useful for our endangered species. So at this point, I'm going to check my time here. Yeah. Okay. Talked a lot about sperm, like I promised. I warned you in advance. You're probably all thinking, okay, that's cool. What about the girls? So this is the other aspect, because have you guys ever seen dog eggs? These are aren't they so cute? Everything I'm showing you in my video, I don't have puppy pictures, but I have really cute gametes. These are dog eggs, domestic dog eggs. These were actually part of my dog in vitro fertilization study. So this is the egg itself, and this is kind of like the eggshell and then the supporting cells. So the sperm actually have to get through a lot of layers to fertilize the egg, but they're adorbs. Okay, so we have good sperm. We're going to continue to improve it. Now we want to do artificial insemination with this cryobank sperm. And now we're back to this figure that probably gave you guys headaches earlier. So this is a domestic dog reproductive cycle. And I showed it to you before with the estrogen and progesterone. What we didn't point out before is that this is really, really long. If you guys are remembering high school health class and the female reproductive cycle, or just are familiar with menstruation, would recognize that normally in a human, ovulation is happening about once a month. In the dog, this is minus 30 days up to 100 days. This is a very long period of time. And when I mentioned to you before that ovulation happens right here, this is the only time this has happened in this period. So in domestic dogs, they're actually only ovulating maybe 1 to 3 times a year. They actually have these really, really long periods where their ovaries are quiescent. They're not doing anything, which actually sounds great. But is not great for our canid reproduction efforts. And this is particularly a big point in our endangered species because they are also seasonal breeders. So this actually comes out to 1 time a year that ovulation is happening. So if we miss that 1 time a year, we've missed that breeding opportunity for the year. And we do not have a really, really clear picture of what the hormones are like in the red wolf or a lot of other canid species to precisely time when ovulation is happening. Again, this is the domestic dog profile where we were able, not we, this is probably a study from the 1990s, but able to take daily blood samples or even multiple times a day blood samples from dogs to track the estrogen change and the progesterone change. And there's also a lot of really nice ultrasound data showing the egg ovulating. We can't do that with red wolves. We have to be really hands-off because they're meant for reintroductions. We don't want to get them habituated to people. And I don't think they would like daily blood draws anyway. So we need a way to be able to create the same profile of hormones, but non-invasively. So ideally through fecal samples or urine samples or something like that, that we can track these same hormones that are circulating through the body, but not do a blood sample and then correlate it with behaviors and pregnancy outcomes with the animals. So this is actually one of the other ways that we're working together with Endangered Wolf Center and a lot of the other institutions in the Red Wolf SAFE program this year, is because with the reintroduction effort, there's been a huge expansion in the number of red wolf breeding pairs that have been matched up. And so this is a really great opportunity to track through the poop the hormones of all these females in the breeding situations and identify which ones, give birth at the end and backtrack it and identify exactly when the best time for that animal's ovulation was and for fertilization to happen. So this is the goal. One of the things we are thinking about while we're doing this also, and I call this dogs are weird, but I switched it to canids are weird because it's more broad than that, is that we do actually have a pretty nice window for when we do decide to do artificial insemination. So we're going to use the hormones, I hopefully really fine-tuned, to figure out through the poop when that animal ovulated. And then we do this thing where we have to identify the best day for that egg to be fertile. And what we found in our work with domestic dogs, or really generations of work in domestic dogs, is that they're really weird. So they actually will ovulate an egg that is not able to be fertilized yet. And this is different from humans. In humans, if anyone's familiar with human IVF clinics, what they'll do is stimulate an egg to grow and then actually pull that egg directly out of the ovary. So they don't have to try to find it in the fallopian tube. They pull it directly out of the ovary, and the process of removing it from the ovary will get that egg to mature the last little bit that needs for it to be able to fertilize. Not what happens in the dog. For some reason, and we do not understand it, dogs will ovulate, or canids will ovulate an egg, and it'll actually take about 4 days hanging out in the oviduct, chilling. And then it will finalize maturation and be able to fertilize. We don't understand this process. This is actually a really cool area of study in the future. If anyone's interested in graduate school and really wants to go in depth in the dog, we need to figure this out because we can't currently replicate this in our in vitro systems. So if we get an ovary right now, we've been cryopreserving the tissue and everything, but we don't actually have a consistent protocol for taking those eggs and maturing them in order to get them to the point where we could fertilize them. So this is one of the big next steps. But what it does mean, and I don't know if I guess this is good, is that once we've identified ovulation, we actually have 4 days to plan our AI, which is better than some species maybe have. Still a narrow window, a lot of moving parts clearly in order to do that, but this is something that we're keeping in mind as one of these quirks of canid reproduction. So with this, we have been asking all these safe institutions to collect poop for us so we can identify these days of ovulation. So, everyone's trash is actually our treasure. We want the feces, please. I'm sure I'm going to get the best nicknames out of this talk. And the other thing we're actually trying to do, and this is actually a big collaborative effort with Karen Bowman from St. Louis Zoo and also Karen Goodrow back at Point Defiance Zoo and Aquarium, is to also address what we can do about understanding why we only have about 25% success rates in this. So right now, we do not know if the reason we only have 25% is the animals are not getting pregnant or if they're losing the pregnancy early. So what we need to do to even try to figure out even try to fix the problem, we need to figure out what's going on first. And for that, what we really need is a way to detect pregnancy early. So not just at the end of the reproductive cycle, but at the beginning. We need something like the human early detection test for canids. And we could probably get them to pee on a stick, right? You've been training them. Yeah, right? Easy peasy. Feel lucky. Not the red wolves. So this is one of the things that is on the horizon. So we're collecting the samples right now. We're going to start trying to identify a couple markers. There have been a lot of work in recent years, again, in other wildlife species and agricultural species, and developing different biomarkers for pregnancy detection that work. They do not have the same system. We can't use the human pregnancy detection test. Everyone's tried, yeah. It will not work. And so one of the candidates, for example, is one that's been used in cheetah. They have the same issue where they really need early pregnancy detection. There's not currently a marker. And they did a non-invasive way. They took poop and looked at all the proteins in the poop and identified ones that were elevated in pregnant animals and not in non-pregnant animals, and found a couple different markers, but one of the ones that they landed on was this immunoglobulin, so it's an immune-based protein that actually was elevated really quickly. This is a Western blot. This is a protein, depending on how bright the band is at the different weeks, is how much protein was present in the poop, and it elevates really quickly after breeding, and actually had another spike in pregnant animals at week 4, which they postulated was actually the time of implantation. So it actually would be a great marker of saying, okay, this pregnancy is continuing on, implantation happened, and it caused an immune response in the animal. So this is one of the markers that we'll be looking for in the red wolf samples to try to develop this pregnancy detection. So in the last half a minute that I have here, I will mention very briefly gonad cryopreservation. So, as I said, we've been freezing samples. We actually will get ovaries and testicular tissue, testes in from animals when they've passed away, or if any reason they need to be spayed or neutered. Sometimes the animals have like a infected uterus or something like that, and they'll actually have to spay them, even though they are young and breedable, otherwise animals. And they will ship us the ovaries and testes, and we will try a couple different cryopreservation techniques that have been developed for other species in our animals right now we have very, very small sample sizes for this, but we're actually in conversations with Karen at St. Louis Zoo about developing this project more in depth in a couple other canid species and focusing really on our domestic dog model to optimize something and then apply it to our red wolves and Mexican wolves and other canid species in the future. So I'm really at the end of my time. With that, I would like to thank everyone for their time, and huge thank you to all of the partners in this work. I always say that any of this research and efforts in the species conservation, it doesn't take a village. Actually, it probably takes a whole city. So Red Wolf SSP and SAFE and U.S. Fish and Wildlife Service, and of course Endangered Wolf Center and St. Louis Zoo for hosting us this week and allowing us to come and collect sperm and seminal plasma from your males and all of our funding sources. So thank you so much.